Hi, I'm Melanie, and I've got something to say. Why are people still talking about thin layer chromatography in 2022? Because it is still a useful tool for synthesis chemists and because it is the fastest and the cheapest way in chromatography for a rapid yes or no answer. So why am I not talking about TLC today? Because TLC is for preliminary results only. And what am I talking about today? I am talking about the other high-performance chromatography, HPTLC, and why HPTLC is the choice for lipid analysis. I would like to show you the results obtained during a feasibility study done together with one of the leading manufacturers of mRNA-based vaccines. First, I would like to give you a brief introduction on the analytical tasks, then explain you the methodology and which instrumental setup is recommended, and then I will present the results. In the second part, I would like to give you an overview about the use of HPTLC coupled to mass spectrometry in the field of lipidomics. The main topic is the analysis of lipids and lipid-based formulations. To prepare liposomes or lipid nanoparticles for drug delivery systems, different types of lipids are needed. And the lipids have a hydrophobic tail and a hydrophilic head. In aqueous solution, this leads to the spontaneous formation of vesicular structures by spontaneous self-assembling. And the manufacturers of lipid-based formulations get the lipids from different suppliers and have to check their identity and purity. And the main task is to test the stability of the finished products and to screen for potentially harmful degradation products. This can easily be done by HPTLC. Liposomes can be used as drug delivery systems for both hydrophilic and lipophilic drugs. So the hydrophilic drugs are encapsulated in the aqueous core and the lipophilic drugs are embedded within the lipophilic double layer membrane. Besides the lipids and the selected drugs, the final formulation contains excipients. So one typical excipient is sucrose used as cryoprotector. Why are some drugs encapsulated in liposomes or LNPs? Fragile molecules like mRNA used for vaccination are encapsulated to protect them against digestion. Without a lipid shell, those molecules would be chopped before they reach their target. So which challenges do we face when analyzing liposomes or LNPs? During chromatography, depending on the selected conditions, hydrolysis processes can occur and then additional degradation products might be formed, which were not formed during the storage of the formulations. And another challenge is the weak retention of polar lipids on reversed phase layers. And most lipids do not show a response in UV vis detection, then other detectors or a derivatization is required. The eluent has to be compatible with the detector used for column chromatography, and for gradient elution, different ionization or in general a different signal response can be caused. How can HPTLC overcome those challenges? And how can QC labs benefit from the use of HPTLC. The most common stationary phase in HPTLC is the normal phase, the unmodified silica gel, and the main separation mechanism is adsorption, an interaction between the functional groups of the analytes and the polar groups of the silica gel. This means that this separation mechanism is complementary to the partition chromatography and can perfectly be combined with reverse phase column chromatography. Another benefit of HPTLC is its flexibility for combination with different detection modes and on-plate derivatizations. The solvent is absent during detection and does not interact. For coupling with mass spectrometry, the selected zones can directly be eluted with a suitable solvent. And last but not least, HPTLC is the only chromatographic technique with a real visual result. The plate itself or the captured images can be inspected by eye, and samples can easily be compared side by side. So the HPTLC chromatogram consists of HPTLC fingerprints, and an HPTLC fingerprint is a pattern of separated zones with a start and end position for each sample track. And non-eluting substances will stay at the application position, but they won't be overseen. So HPTLC is the perfect cross-check method in combination with LC methods, as it allows to really see all zones present in a sample. The chromatogram shows the entire composition of a sample. On the next slides, I will present you the analysis of lipids used for formation of liposomes and lipid nanoparticles. The HPTLC methodology is simple. Between 15 and 20 sample tracks can be applied as bands onto one HPTLC plate without cleanup or enrichment. And after development, the plate is sprayed with a fluorescent dye and the fluorescence is measured in the detector. The state-of-the-art instrumentation is the fully automated HPTLC PRO system. And here you can see the steps. The samples can directly be applied. All samples and standards are placed in the sample rack of the HPTLC PRO module application, which can hold up to 75 vials. And the plates are loaded into the module plate storage. Up to 5 plates can autonomously be handled by the HPTLC PRO system without manual intervention. Once the system has started, the samples are applied onto the first plate, and as soon as the first plate is developed in the HPTLC PRO Module DEVELOPMENT, the next plate enters the system and is applied in parallel. Then the next step is within the system is the automated derivatization by microdroplet spraying technique. After this step, the plate is detected. Currently, detection is done by capturing images at different illumination wavelengths with the documentation system or by scanning densitometry. And the module detection for the HPTLC PRO system is not available yet, but will be based on hyperspectral imaging and is expected for 2023. In addition to the classical UV-Vis detection, selected zones can be transferred to a MS with the help of the illusion-based interface for HPTLC-MS. So the HPTLC PRO system consists of the modules plate storage, application, development, derivatization, detection, and MS interface. The module plate storage is for automated feeding of the system with clean plates and for receiving back the processed plates in a separate compartment within the module. The module application uses a spray-on technique for application, and the used microliter syringe is rinsed with 2 rinsing solutions between each sample application. The module development features a new design chamber geometry with low chamber volume for a better control of the gas phase. In HPTLC, besides the liquid mobile phase and the solid stationary phase, the gas phase, as a third degree of freedom, is influencing the separation. The gas phase can be actively circulated prior and during chromatography and can be used to further optimize the separation. Because of the optimization and the optimized chamber geometry, reproducible conditions for development are guaranteed for validated methods. The module derivatization uses a piezoelectric spraying technology. The formed microdroplets settle homogeneously onto the plate, and the nozzle used for spraying is automatically cleaned after each spraying step. A heating plate is also included. The module detection will use hyperspectral imaging for the UV-Vis range, and the module MS interface, which is also expected for 2023, will allow to select zones of interest via the VisionCat software and directly elute them using the integrated elution pump. The eluate is transferred into a MS or can be collected for further analysis offline. For the analysis of lipids, the total time required for 5 plates with the steps from application to detection is about 4.5 hours. If 20 tracks are applied on each plate, a runtime per sample of less than 3 minutes is achieved. In this short video, the entire process is shown, including the MS interface. So what do we get as a result? We get, as mentioned before, an HPTLC fingerprint for each sample or standard track, showing the entire sample composition. Nothing can be missed and results can easily be interpreted by eye. For quantification, profiles from images, scanning densitometry or hyperspectral imaging are evaluated with the VisionCat software. The 2 figures show the fingerprints of lipid-based formulations at long-wave UV light after derivatization with Primoline and at white light after derivatization with copper-2 sulfate. So why did we choose Primoline for the derivatization of the lipids and the formulations? Primoline is preferred for the formulations because sucrose, one of the excipients, is not derivatized. You can see this here at the application position with a lot of reaction caused by the copper-2 sulfate. So in general, we get a better background for Primoline. Furthermore, Primoline allows detection in fluorescence mode. In comparison to absorption mode, fluorescence mode is more sensitive, has a better signal-to-noise ratio, and allows for evaluation in a wide linear working range. And Primoline allows for direct coupling with HPTLC-MS because this fluorophore does not interfere with the mass spectra of lipids and gives a separate signal. Now it's time to have a look at the results. So in our feasibility study, we obtained a good separation and signal response of the most common lipids used for production of liposomes and lipid nanoparticles. And the purity and identity of the raw materials can be determined with the calibration curves generated within the VisionCat's software. For the liposomes and the lipid-based formulations, the same methodology can be used. For stability testing, the formulation was applied without sample preparation or dilution step, and the liposome solution was diluted 1:6 for determination in the same working range. Interestingly, the excipients like sucrose do not disturb at all the separation and detection, and the RNA in all excipients are either not derivatized or they stay at the application position. The chromatogram also shows a stressed formulation, which still does not show any degradation product. And for the quantification of the different lipids, this is done by multi-level calibration. As examples, you can see here the calibration curves of the 3 lipids, DOPI, DOPES and DODMA. After discussion with the manufacturer, a second approach was developed which allows to separate the known impurities or degradation products within the formulation. The oleic acid is not sufficiently separated by one-step separation, then it's here on top. So in this case, a rapid 2-step development is done in the module development, which increases the total runtime just about 5 minutes per plate. So for 20 sample tracks, this is not really relevant. Instead of one development up to 70 mm with chloroform methanol water, the plate is only developed up to 30 mm and then developed with a second solvent mixture of another selectivity up to 70 mm. Yeah, and by this, the oleic acid migrates up to RF 0.45 instead of 0.8. So for whom is this setup and methodology of interest? It is of interest for all pharma and life science companies as well as all lipid research groups. So let me conclude. HPTLC allows an easy, rapid and robust high-throughput testing of complex lipid-containing samples. And with the HPTLC PRO system, the processing of 5 plates can be done without human intervention, even during the night. The analyst can rapidly analyze and evaluate those 5 processed plates whenever feasible. An application note on the shown results can be downloaded from the CAMAG website. HPTLC is a highly flexible platform for multiple detections. Besides the classical UV-Vis detection of native and derivatized substances, effect-directed tests can be done. The silica gel allows to perform enzymatic assays and biological tests on the plate after chromatography and therefore to couple the chromatographic separation with activity tests. Is a substance present with a desired or unwanted effect, e.g. a genotoxic effect? But in the second part of today, we will not discuss about this. We will talk about the hyphenations and especially hyphenating HPTLC with mass spectrometry in the field of lipidomic research. There are different possibilities for direct HPTLC-MS coupling, like HPTLC multi-MS, and here you can see an elution-based approach. The elution head of the MS interface is moving to the selected position and is lowered for elution. The valve switches from bypass to injection position. And to prevent fouling of the ion source with silica gel, an exchangeable filter is installed. Each elution takes about 1 to 2 minutes. How can we select the zones of interest? In the detection step, all zones of interest from different detection modes can be selected for elution using the VisionCat software. And the lipids are best detected with Brimulene in fluorescence mode And in the MS, the primoline and the lipids will individually be detected because primoline is not covalently bound by the lipids. So here you have signals from primoline and the lipids will have another mass spectrum. Primoline is a mixture of oligomers of substituted benzothioazoles. It does not interfere with the mass spectra of lipids. What are the advantages of HPTLC-MS? HPTLC is an offline technique and the developing solvent is evaporated before mass detection. So this allows to change and to an appropriate solvent for mass detection. No gradient for elution of selected zones is needed. So the eluent for mass detection has a constant composition and is in this way similar ionization efficiencies are obtained for the individual lipids in each class. The entire sample is loaded onto the plate, so all degradation products or other changes in the pattern of the chromatogram are immediately seen. And where can we use HPTLC and HPTLC-MS in the field of lipidomic research? There are many different application examples, and different application examples were published in the last decades, like detection of monoacylglycerides in biodiesel, heavy petroleum products analysis, analysis of sphingolipids in human plasma, phospholipids in different biological matrices. And this review article published last year from Vicente Cebolla and co-authors gives a good overview about the current state of lipidomics and application for HPTLC-MS. It also describes different coupling possibilities. The general review about lipid analysis by HPTLC, written by Fuchs et al., is a good source of methods for the separation and analysis of different classes of lipids. I'm running out of time, so let me briefly summarize why HPTLC is the other high-performance chromatography. Because it is absolutely complementary to column chromatography and allows for a rapid parallel analysis of many samples. HPTLC leads to a separation in location instead of time and offers the possibility to analyze the entire composition of a sample. The obtained two-dimensional array of separated substances is detectable many times, which means that we are able to gain multivariate data from one separation in different illuminations and by using different reagents or even after incubation with enzymatic solutions or bacterial culture media. You can find a lot of more information here. I highly recommend our literature database, CCBS, with more than 11,000 abstracts from TLC and HPTLC publications. Furthermore, you can find applications in the CBS journal and download application notes. Also, several webinars are available. And for direct contact, please write to request@camag.com for application support, to sales@camag.com for information about instruments and software, and to service@camag.com for technical questions. A final thing to highlight is the HPTLC Association. Starting this spring, every 3 months, a HPTLC online user meeting will take place. More information can be found on the blog of the association. Thank you for your attention.